So I thank the organizers about this beautiful conference. And now we're going to do some preparatory stuff for these talks that will occur in December. There will probably also be a third talk on this using this mapper software, which we had discussed in class in August. But since that was August, we will now review the mapper software, reviewing first this paper on extracting insights from the shape of complex data using topology. There are many ways of analyzing data using topology. One of them is this mapper software where the input is basically a bunch of data points. And these data points could be data points living on a hand, or they could be data points representing protein shape, or data points representing DNA sequences. So they can represent a lot of different things. In the mapper software, one uses a particular one wants to maybe analyze a particular function. So maybe if it's protein, you want to analyze the energy of the function, or maybe it's data and you don't really know what you want to analyze. So maybe you'll do a principal component projection. So you might want to project your data onto, for example, onto R. Oops, I forgot to, I'm forgetting all my standard things. So you might want to project your data onto R, study something. So in this particular very simple example, we're going to just project onto the x-coordinate. So we're going to use the x-coordinate to filter our data. And then we use this x-coordinate to partition our data. So we now have this map that goes from our data points, just projecting onto the x-coordinate. So we have it projected onto R. We'll then take pre-images. So we'll divide up the real line into intervals, take the pre-image, and then we use that to then partition the data. So we will bin the data into this pre-image of an interval. Then we've got this pre-image of an interval, so that maps up to here, and we bin that into here. And we do that for a whole bunch of intervals. Just constantly take this pre-image and then bin. I have to remember to hold the pencil properly so I don't actually touch it. Okay. Eventually, I get my technology sorted. So just continue taking these things and breaking your data. Now this data is in 3 dimensions. So when we bin into here, this is actually, the thumb is separate than the hand. So the step 1 was take your data. Step 2, decide some function of interest. Maybe that's energy, maybe it's the x-coordinate. And then cover your real line. So we cover this real line with intervals, take the pre-images to separate it, and then we cluster each bin. So each bin is clustered. So a vertex, so for each connected component, we get a vertex. This is a connected component. In 3D, the thumb is separate. And so we get vertices for every single cluster. And then so we look at the bins separately. So the clustering is done separately on each individual bin. So each individual bin is clustered. After we do that, we then will say, suppose there is a non-empty intersection. So if we take a look back over, we can see that this red here, that there's an intersection with this red here. So there were some data points in common in terms of, so we draw an edge. So whenever there's an intersection, we draw an edge connecting. So this cluster right here intersects with this cluster here, so we draw an edge, intersects with this cluster here, so we draw an edge. And so eventually we get this graph that basically preserves the shape of our data to some degree. So that is the steps. These steps are, you start off with a bunch of points. Your points may represent proteins, they may represent data points, sequences, whatever they represent. Then choose any function of interest. So any function of interest, maybe energy, maybe projecting onto the x-coordinate. Put the data into overlapping bins, and then cluster each bin, and then create the network. And we can see that with our excellent choice of projecting onto the x-coordinate, we basically recover the shape of the hand. So we went from thousands of data points up here to only 13 data points, and we can see the shape of the hand. There are, however, a number of choices that we made. One of the choices, probably the most important one, and this is why the program is called Mapper, is your choice of the map, the choice of this function here, will depend upon, will affect what you see. If we had instead projected onto the y-coordinates, we wouldn't have seen a very good picture of the hand. So choosing your function here is the most important choice, and that will definitely affect the output. But one can make a variety of different choices and analyze a variety of different functions to see what works best. Then the other choice that we had to make, so this was the first choice and the biggest choice. The second choice is, what's the size of our bins? We're now basically covering R, so we projected onto R and then used some sort of cover of R. So what's the size of our bins? What's the shape of our bins? That's a choice as well. Generally speaking, because there's a choice here, this one's not too bad. What you can do is just run the software multiple times using a variety of different bin sizes, and then see if you get the same result. If you get the same result, then that means it's probably a valid result. If making minor changes into your bin size, well, then maybe your result is not as meaningful. So choice number 2 is not so bad, because you can check a variety of different choices and see if it persists throughout those. Then the third choice is we have to choose some way of clustering. There are lots and lots of different ways to cluster data. And so this can, again, have an effect. And frequently people, again, run a variety of different choices in terms of the clustering. So you might run a few different things in terms of your clustering as well to see if you get basically the same result. But that is 3 different choices that need to be made. I like my old research on tangle analysis because everything was so clean then. You had your model, which the biologist gave us, and then we just did the nice math, and it was nice. But here there are choices that need to be made, hopefully in collaboration with the people who generated the data, so that you make good choices, especially with respect to choice 1. The other ones you can do a variety of just run the software multiple times. So mathematicians can probably do 2 and 3, but with respect to clustering, understanding the biology, and even deciding the bin size, understanding the biology can help quite a bit. So any questions with regard to how this software basically works? One can modify. We did a simple example where we went into R, but you can choose your function to go into any space that you want. And so if you've got 2 functions of interest, you can map your first coordinate onto R1, your second coordinate R2, and then take a cover of R2 instead of a cover of R1. You can map onto any space you want. If you want to map onto a torus, if you want to map onto a Klein bottle, whatever you want to do, all you have to do then is choose a cover. And there are different covers that you can choose. So in the last one, we chose rectangles or squares. In this one here, we can choose polygons instead. And you'll notice that in higher dimensions, when we do the intersection, we can actually, when we map back, so if we map back, maybe this will map back here, this one will map back here, this one will map back here. And we then cluster, so let's put a few clusters down here. In this case, everything clusters into just one single thing. We then said, remember, our data points are our clusters. If 2 of them intersect, so they intersect here, we draw a line segment. 2 of them intersect, we draw a line segment. 2 of them intersect, we draw that line segment. We can also, instead of just drawing a graph, if 3 of them intersect, so we've got 3 of them intersecting, create what's called a simplicial complex and actually get a higher dimensional structure as well. And so one can do more things than just create a graph. But most of the examples, I think all the examples published so far, have been using the graph, but one can do this higher dimensional stuff as well and see what appears. So there's lots of different choices in choosing the map and choosing this filter. It's good to understand the biology problem. Any questions there? So we now want to take a look at a particular example. So our main thing that we will take a look today at this paper out of the Carlson lab on methods for exploring low-density states and biomolecular folding pathways. The particular example that they looked at was this RNA hairpin, and they analyzed specifically the secondary structure. So they looked specifically at the secondary structure and the folding pathway of the secondary structure. But as long as one has a distance between shapes, and there's lots of ways of defining distances between shapes, they could have instead done the three-dimensional pictures instead. So in this example here, basically the data was contact maps. So that means they knew the contacts between the RNA bases right here. So that was the data from a particular type of molecular dynamic simulation of this GCAA tetraloop. And they did use the folding at home. So if you don't have enough computers to do your folding, maybe you can use the folding at home software to generate your data. Or better yet, just work with somebody who has data. By the way, there are students in my course who are looking for data. So if you would like your data analyzed by either me or one of these students, some of whom are graduate students, others of whom are professors, let me know. We would love to get some data. So in this particular case, they broke up their data into 760 trajectories that showed the unfolding and 550 trajectories with the refolding. And the goal was to determine the pathways using the secondary structure between the folded and unfolded states. So there had been hypotheses that there were two main paths and other hypotheses that there were multiple paths between the folded and the unfolded states. So what the problem though that they have in terms of analyzing these things is when you run these simulations, the intermediate conformations are of rather low density. Their data is actually not very good. In fact, they warn the reader that the data actually had not yet reached equilibrium. So their data, they actually need to run this a lot longer even for this small conformation. But once you have the data, then you can run the mapper software. But the data here is a little bit suspicious. It has not reached equilibrium. And even if it had the intermediate conformations, there's a lot fewer of those. So how can you distinguish the intermediate conformations from just plain noise in your data? So if you don't have very many intermediate conformations, how can you identify them? Well, their solution was their filter function was to go from the space of conformations, and their space of conformations was basically what contact. So this would be a conformation if we just specified that these two contacts were together. You can instead have your space of conformations be your 3D coordinates of each individual atom if you want. That's actually not a problem for the mapper software because all we need is a distance. So if you've got 20,000 data points, 20,000 atoms, as long as you can tell me a distance between your protein 1 and protein 2, that's all I need. And so it's that distance, where that distance comes from, if it comes from simplifying things and using a contact map and a Hamming distance, which is contacts, which counts the differences between the contacts, between the different forms, doesn't matter to us where it comes from as long as we have the distance. So as long as I get a distance between structures, we can go ahead and use the mapper software wherever that distance comes from. And it can even be a fuzzy distance as well. It doesn't even have to be all that well defined. So they went from the space of conformations, these contact maps, and outputted the density. That way they could filter. So if we take a look at a particular example, they could filter based upon the density. So for example, all of these structures here were not the linear one, so this is the folding pathway. The unfolded structures there were many, many more of those structures than there were these structures. But if you filtered it and binned these into a single thing and then said, we're only going to look at these structures, then among these structures, what is high density and what can we cluster? And so they clustered. So there were 23% within this family, 44% in this family, and 3% within this family. So by using this density filter, they could look at the low density, so that they didn't get confused between the vast majority were unfolded. They could then see these stuff right here. Keep in mind that this is an entire family of structures. So we are clustering these things and one representative is drawn. And then you can kind of see there's this dotting here. So they're showing some other representatives. So this structure actually represents a family of structures. And you can kind of see that a certain percentage. Unfortunately, even if I really blow it up, so I think I can kind of see that there's 71% have this contact, 72% this one, 57% that. I think, you have to really blow up the figure to try to read these things, but it is a family of structures within each of these bits. So any questions there? I will very quickly show, so this was 550. Yes. Are you talking about generating the data, or are you talking about analyzing the data? So basically, the more programs out there, the better. But I did not mention the advantages. I actually cut those slides from my August one, and I should have left them in. One of the advantages is that when you're doing the topology of this sort of thing, noise is pretty much, it gets binned within here. So you don't have to worry quite as much about noise in terms of the clustering because it kind of gets binned in. That's probably very similar to the UCLA stuff in terms of handling noise. Most programs these days are pretty good at handling that. But we can work with any type of filter function. So you can analyze it from a variety of different perspectives. They did specifically density. So with respect to specifically density, I'm not actually that familiar with the UCLA, but do they output a complete graph, where you have connections in terms of showing these different things? Does the UCLA also give you this sort of graph type stuff? Yeah. So one of these things is the graph is very helpful because with the graph, you can see that 23% go in this pathway versus 44% in this pathway. And then there's some noise here. The 3% actually represents the reptation type of thing. The reptation is where you make a contact and then this contact moves along here and then moves along and moves along. And that reptation type thing seems to be specifically noise. So if the UCLA one does not determine, a graph and it only determines a tree, a lot of these things only determine trees, then you don't see that there is more than one way to get from the beginning to the end. So this is one way for multiple pathways. That may or may not have done a good job answering your questions because I'm not actually too familiar with the UCLA software. So that, any other comments on? So we can see for the folding pathway, there are these two different pathways, and they looked at 550 trajectories involving about 3,000 conformations. Truth in advertising, I'm going to very quickly go through the math. This is actually the filter function that they used. It basically assumed that your density function had followed a Boltzmann distribution of things, and we looked at, in one case, you looked at the refolding events where the majority of your conformations And so if we take a look at the coloring red equals to your densest region. And so densest region. So when you start off with an unfolded, that's the majority of your complexes are red. The majority of complexes are just linear. And then your least dense goes down to least dense, goes down to your blue. And so while there were 550 complete refolding things, we saw a lot more of the linear stuff than of the nonlinear stuff. In terms of the binning was each of the bin sizes had exactly the same number of elements. And so you can basically take the 3,000 and divide it between our 8 bins right here. So we had 8 bins of equal size. And since they are of equal size, we can see that the first 4 bins had basically only the linear structure. So about 40% of the data was purely linear. Anything that was less than 100% was not shown. Anything that was less than 3%, 100%, 99%, those are all good values. But if it was less than 3%, that is not shown. So for example, this says 99%. So that means there's a 1% thing where there was not, that was not linear in this thing here. But here, 98% were this, 100%, and then this one here was the only one that the clustering broke up into 3 different shapes, one of which did not go on, the reptation model, which did not go on and complete to fold. So any questions there? One of the interesting things that they found was that in the I haven't gotten there yet. So one question they had is, how real is this? Are these 2 paths? Well, one of the things they can do is go back and look at the data. And so actually, let's go back and say what we need to do with this data. What they then did is say, let's combine, all of these are unfolded anyway, so we'll combine this into a single unfolded node. This is partially folded, so we're going to go ahead and combine this into a single semi-folded node. And then we'll compare if we go this pathway or this pathway and take a look at that. So that's what's on this next page, is we combined all the unfolded into this node right here, the partially folded and folded into this node, and then the other 2 pathways, so where we had this going this way versus this way. And so these 2 nodes are those intermediate ones. And to see if they really are real pathways, or maybe you actually go between them, they looked at their data to see what was the probability if you are within this node right here that you go back to being unfolded. Well, that was 18%. If you go back to being if you go to actually being folded, so going forward and being folded, that would be 19%. 62% of actually staying within that conformation, and only 2% of going into the other one. So there was very little exchange between these 2 conformations. So since there's so little exchange, it looks like there really are 2 main pathways to get from the unfolded state to the folded state, where one can go either this direction or that direction. And of course, being biology, one doesn't always travel forward. One, goes back and forth a little bit. Stays in here within. And by staying in here, any questions there? The unfolding pathway is actually very different than the folding pathway. With the unfolding pathway, it basically is just this linear pathway right here, where we start folded and we go unfolded. You can see here, there's actually only 81% was in this structure right here. And there were a bunch of other small clusters, each which had less than 3%. So there were some other things going on, but if it's less than 3%, that could be just noise in your data. The main thing with respect to unfolding was a unique pathway. So there was quite a bit of a difference between the folding and unfolding. Folding, there seemed to be two different main pathways versus unfolding is exactly one pathway. Any questions about that? Okay. So I won't talk about this data, but there is a variety of other places where they've applied this mapper software. They've applied it to breast cancer data and found some interesting substructures there. In this case, they actually, in this EQTL Biological Data Visualization Challenge, they won one of the best visualization things for this challenge right here. And this is where they were analyzing SNP data. And so they clustered single nucleotide polymorphism. So the data in here were data points where, again, you clustered a number of different SNPs. And then if there was intersection between things, you drew the node. And so this is the structure that they saw. And then they color-coded the structure where blue was disease-free and red was disease. So you can see that where it is red, the SNPs more correlated with disease. And you can kind of see where the correlation with disease is strongest. And where some SNPs, these family of SNPs don't really correlate with disease at all. So it is a nice way of visualizing data, whether it be protein data, RNA data, or gene sequences, etc. As long as we've got a distance, and it's one way of analyzing. Any questions there? Okay. I'm going to also, I actually started this one on Wednesday, but I only gave the background that all of you guys at the conference already know. But I was hoping to also finish off this one to prepare for Yusu's Wong talk on Monday, December 9th, which is basically just visualizing landscapes like the topological energy, the energy functions. And so to review what we did on Wednesday, and which all of you guys in Banff, Canada already know, an energy landscape is basically just mapping your set of conformations. So you've got our set of conformations here and mapping them to the energy. So if I've got a conformation here, it would get mapped to, say, this energy here. So this would be mostly unfolded versus something that is more folded would get mapped to a lower energy. And so we've got this energy landscape, which we'd like to visualize. But the problem with visualizing energy landscapes, if we take a look at this fairly simple example, the protein that they looked at was called survivin. And basically, they believe that if you take a look at the protein conformation space, it's 20-dimensional. You know, I don't know about you, but I have difficulty visualizing things in 20 dimensions. And so the idea is there a better way of visualizing the landscape thing when you have something that is high dimensional? And it reduces to something 20-dimensional. For example, if I have a circle, I have to draw this circle in 2-dimensional space, even though it is a 1-dimensional object. So when I say 20-dimensional, this actually might be in something that's 300-dimensional or 5,000-dimensional. They actually looked at something 1,000-dimensional, although you could have reduced that by quite a bit. But the actual shape is something that's 20-dimensional. So how can one visualize that? Well, one way to visualize this, and this is an old thing from the 1940s, is the contour tree. For the mathematician among you, if you have a nice function, this is the same thing as your Reeb graph. Except for this example, we're going to take a look at simply connected domains. So we will get a tree, no circles, although this paper I'm talking about is from 2010. There will be serious updates in the 2013 talk in December by Yusu Wang. So in the Reeb graph, you basically have a filter function. So in this case, our function goes from our space of conformations. So this is for the case of survivin, some 20-dimensional space. We go to our energy right here, And then we can take the energy and take the pre-image of this energy, and that pre-image we just map to a single point, if it's connected. If it's not connected, if I take the pre-image of this, so let me take the pre-image of this one, the pre-image of this one decomposes into two components, and so we would get 2 points for that. And where it joins up, so where it merges from two components to one component, so everything below this line is two components, everything above is one, we then merge these two. And so basically we're just doing, for the mathematicians, the quotient topology. For the non-mathematician, just squeeze all these components. So squeeze these components, so all of this gets squeezed to a single line, here, and then this gets squeezed to a single line right here, and then this will get squeezed for a time to a single line, and then this part here and this part here also gets squeezed to a single line. So that's the contour tree. That's an old idea from the 1940s that has been used mathematically, called the Reed graph. One would like to get a bit of additional information though, because for one thing, if I take the pre-image of this edge right here, so we take the pre-image of this edge, it gets mapped to this thing right here. Let me actually clear off my screen. So if I take the pre-image of this brown edge, it gets mapped to here. If I take the pre-image of the blue edge, you know, that blue edge gets mapped here. If I take the pre-image of this one, it gets mapped here. You'll notice that in this case, it's drawn as area, but in reality, we have a 20-dimensional manifold, and so this will get mapped back to some volume. But this volume is much less than this volume, and we'd like to be able to visualize there's a lot less of this than there is of this. So what you can do is whatever this volume is, make that the length. Whatever this volume is, make that the length. Whatever this volume is, so we take a look at the pre-image of this thing right here. We get this brown thing right here. Whatever that volume is will determine the length. So that's been around since the 1940s, a nice way of visualizing energy landscapes. But whenever we saw these energy landscapes, you know, it was nice to be able to see these nice, you know, three-dimensional pictures. And so what we can instead do is we'll define CQ to be the contour in M that is collapsed to Q. So this entire edge, if I take the union of this, this will be the topocomp of the edge. So the pre-image of this is the topological component. It'll be all this brown stuff. So the pre-image of this edge, this is the topocomp of that. The pre-image of this, we have the topocomp of that. And so one thing one can do is to try to come up with a map instead. So originally we had a map from our 20-dimensional conformational space into R. We want to instead come up with a map from two-dimensional space into R. And the reason we choose R equal to 2 is because then we can take a look at the graph. And so our domain is R2, our output is R. And so if you have a function from R2 into R, you can visualize the graph in R3, and that just gives you a nice visualization. So the idea is to find a function that will be related to this function right here. And the properties that you want it to have is you want it to share the same contour tree. So it should share the same contour tree. And the area, if I take the area of the pre-image of an edge with respect to G for the contour tree, I want it to be the same as the volume if I take the pre-image of an edge for F instead. So this method, by the way, expands upon Weber's topological landscapes, published in 2007. So let's see a generic picture. Taking a generic picture of this, we basically, the algorithm would be to first come up with your contour tree. So we take our 20-dimensional configuration space and create a contour tree. We can then go from our contour tree and create this map G from R2 into R. And the basic idea is all the points along here should have the same energy. So they'll get mapped to something. And so we will just choose a curve in R2, and we will see, and this choice will be arbitrary. So this pre-image, which will actually be some 20-dimensional object, will get mapped to all having the same energy over here. Because we're in R2, we're going to map it to some curve right here. And that curve, it'll be an arbitrary curve, but it will be everything that gets mapped to exactly the same energy. And the condition, there's only one condition we're going to have about that curve, and that curve, the only condition is that the area that is contained within this curve will be the same as the volume. It'll be the same as the volume of everything below it in the graph. So since we already have this, we can take a look at this, and whatever the lengths are this, that adds up to the appropriate volume. We will say that that must be the area. And then as you go down, let me clear my picture again. As we go down into this thing here, we can then take a point that is closer. That'll get mapped to this, so it'll have less volume because this corresponds to less volume. When we get to a split right here, or as we get to the split here, we would get some kind of figure 8 structure that would then have the volume of everything below. As we go down, we would have a contour here that again has less, volume, so less volume here. And so that's the basic algorithm. The algorithm that was implemented used rectangles, and you can use any shape you want. And they implemented 2 different shapes. I kind of like the rectangles because, remember, this is just a visualization. And whenever you have a visualization, you do lose information. So the fact that it looks a little artificial, I actually like because it reminds me that it's just helping us to visualize the energy landscape. But it is, just a visualization of this thing. So what they'll do is they'll take this point, they'll have an arrow, and this arrow tells me that C of B is contained in C of U. So if I take the data points that correspond to C of U that all have the same energy, that would be this thing right here. They all have the same energy. C of V, it's going to be just the leaf. So it'll just be this one point, so the ending node. So it goes within here. It could be either a maximum or a minimum. So if it's a local minimum right here, we can draw it below. Or it could have been a local maximum. And so in your energy landscape, it might have been a local minimum, or it could have been a local maximum. In which case your C of V would be down here, and this would be higher energy, or it could be C of V is here, and this could be lower energy. But all of this maps to the same energy, and this maps to either a higher energy or a lower energy, depending upon whether it's going down or up, so whether it's a local max or a local min. Any questions about this rectangle? We can then have a more complicated configuration. Instead of just going to a leaf, we still might have that C of V is contained in C of U, but there might be things below C of V, so we still have C of V contained in C of U because the arrow points down. So we have the rectangle with the appropriate area corresponding to the volume of everything below, and then we draw another rectangle with the area of everything below here, and we can continue partitioning. So if this goes into other leaves, we can continue partitioning these rectangles. And one can do this either using, so this is an example on the left using rectangles, and this is an example on the right using a Voronoi tree map. So there's different ways of partitioning these things. So any questions here? So the output basically looks like this, but I actually haven't given you quite all the information. And the question is, where do you start from? In this particular case, blue is low energy, so we do have blue equals to low energy, and red equals to high energy. And so in the upper right, the blue was chosen to be the root of the tree. It's not obvious which is the root of the tree. So you can choose the blue to be the root of the tree, so the lowest energy, or you could choose the highest energy to be the root of the tree, or you could choose some intermediate stage to be the root of the tree. Your contour trees often look something like this, and it's not always clear what is the root, and so maybe you should choose your root to be here, maybe you should choose it to be here, or maybe it actually doesn't actually occur at a node, maybe you put it at some intermediate stage. And one of the reasons to do it at some intermediate stage is now we can look at a collection of families of these maps right here. We can take a look at slowly perturbing this root and taking a look at trees where we put the root here, the root here, the root, just continuously move the root in this direction, move the root in that direction, move it in different directions, and then compare them. And we get is this map here in the upper left, which I've blown up here. You can see, depending upon where you choose the root, in the bottom left, you can see that is where the lowest energy is the root, and so that has the most volume. Over here, the highest energy, and over here, which is very likely the best model of the energy, appears in this diagram right here. And so you can see how it transitions between the two. And yeah, there is an interesting outlier over there, but you can visualize actually all the different energy landscapes depending upon where the root is. And if we take a look at the one where, so this is the one corresponding to this one, right, this object right here. If we take a look at it, that is the energy landscape that we get right here, where there's an intermediate conformation, and you can see the high energy here, the low energy here, and this is a look from the side. So if we take a look from the side, what it should look like. Any questions about visualizing topological landscapes? And this, by the way, applies to any scalar valued function. So we just had a function from our conformations to R. You can do this visualization, and the software is accessible off of Yisu Wang's website for visualizing any scalar, any real valued function. So, any questions there? Okay. Well, we only actually have 30 seconds left according to an accurate clock, so I can remind the students that next week is Thanksgiving break, so there'll be no lectures next week, but we will be meeting 1 week from Monday to talk more about lectures that will appear in the December workshop. Any questions before I sign off? See you in about a week. Yeah, for this topological landscape one, for the 2010 version, it works only on trees. I don't know what the 2013 version is. Since I said I was going to post it on YouTube, she didn't want to tell me, what the stuff actually is. So you'll have to go to the December talk to find out if there is an update that will allow for non-trees. I don't know if the version 3 years later, but this version for the topological landscape is specifically trees. The first part of the talk, any function will do. So, but for this one, it was specifically real valued functions. My guess is it's probably still real valued functions, but maybe it will allow for things other than trees. I'm not sure. Yeah. They're probably the same thing. This is used in a variety of different fields. I'm not familiar with the terms, I can't guarantee it, but it is used in a variety of different fields. So, yeah, and I think I actually do recognize that term. I'm very bad with names, but yes, I think, yeah, because we just saw, yeah, yeah, yeah. It's used in many different fields. These are very useful. And now one can do a 2D representation of it instead. Other questions?
This fellow from the whale, the baby whale, that the physics, the chemistry department, there's a whole book on that. And so it provides birthday because also this connected. Now the chair of the department.
Thank you.
So I think with this nice